Now we are going to discuss about gibberellins. Gibberellins are one of the phytohormone and it is a plant growth promoter. The plant growth is promoted by the gibberellins. It is a second type of plant growth promoter. Here the gibberellins were firstly identified by different scientists, but here first the Japanese farmers identified the extensive growth of rice plants. The rice plants are showing the extensive growth, but no flowering. The flowering is not occurred. That's why here that type of disease is called as foolish seedling disease. So it is due to the production of one hormone or one chemical. So the chemical was produced by one fungus, that is Gibberella fusicori. Next, later so many scientists gibberellins were identified and extracted from different plants. Here first of all, Yabuta scientists. Yabuta scientists he extracted the gibberellins, and here different type of gibberellins were identified and extracted. So for the gibberellin synthesis, the precursor, so the starting material or the precursor is acetyl coenzyme A. So it is the precursor for the gibberellin synthesis. Next here about 100 types of gibberellic acids are extracted from different plants and fungal members. So the fungal members and different type of plants, higher plants like angiosperms and gymnosperms, these were extracted. So they are named as GA1, gibberellic acid 1, as it is acidic nature, it is called as gibberellic acid, commonly also known as gibberellins. GA1, GA2 and GA3. So many gibberellic acids are identified and named as GA1, GA2, GA3. But the first identified and extracted gibberellic acid is GA3. Here on GA3, most extensive studies were carried out. GA3 is the first identified gibberellic acid, and on this phytohormone, most studies, most of the scientists studied about this gibberellic acid 3. Next, physiological effects of gibberellins. The physiological effects are the functions of gibberellins. First of all, stem and leaf growth. So how the gibberellins are showing the effect on plant? Mainly stem and leaf growth. So the stem generally it is showing elongation. So it is a stem and root system. So mainly here the gibberellins are showing the effect on stem and leaves, mainly on aerial parts, not on the underground parts. So the gibberellins do not show any effect on the roots. So no effect on the roots. Stem and leaves are here aerial parts. Here this type of internode, it is called as internode. So the internode is showing the elongation due to the effect of gibberellin. So a rapid elongation, or mainly the stem elongation. Stem is showing the growth. Next, leaf also expanding due to the gibberellic acid, the leaf expansion. Stem elongation, leaf expansion, but gibberellins do not show any effect on the roots. Here bolting is the nothing but it is rapid elongation during the flowering or before flowering. It is a stem, but before flowering it is very short, and nodes and internodes are also very closely arranged. So it is condensed stem. On this stem, a number of leaves are arranged or produced very compactly. So this type of growth is called as rosette type of growth or rosette form. So mainly cabbage, and carrot. So this type of plants are showing here rosette type of growth, or before flowering, when the flowering is not started, so the plant is in rosette manner, condensed form. So a number of leaves are arranged very closely, so compactly arranged. That's why it is appearing like a rose flower manner. So in the rose flower, how the petals are arranged closely, compactly, here also the leaves are arranged on the stem. So it is also called as a rosette form. Here during the flowering process, here when the flowering is initiated or before the flowering, here the stem is rapidly elongating. So it is showing the rapid growth. So it is elongating rapidly. So this type of elongation is called as bolting. Bolting is a rapid elongation before the flowering or during the flowering process. So that's why here the leaves are arranged very distantly. So this type of process is called as bolting. So what is the reason for the bolting? Here the gibberellic acids are produced during the flowering process or before the flowering, when the flowering is started. Next effect is seed germination. Seed germination. In the seed germination also the gibberellins are showing the effect here, mainly in the barley seeds. So it is a seed, here monocot seed or barley seed. In the seeds, here in the monocot seeds only one cotyledon is present. It is embryo. And completely it is endosperm. Here during the seed germination, here the gibberellic acids are synthesized in the embryo. So these gibberellic acids are here transported to the aleurone layer. So the endosperm is covered by aleurone layer. It is a proteinaceous layer, nothing but it is the outer layer of the endosperm. Gibberellic acids are transported to the aleurone layer. Here one enzyme is produced, alpha amylase. Here alpha amylase enzyme is produced in the outer layer of endosperm or in the aleurone layer. It is transported to the endosperm. Here the food materials are converting, or the starch is converting into your glucose. It is involving in the respiration. So in the respiration energy is produced. That energy is utilized by the embryo and it is germinating. So here in the mainly in the seed germination, the gibberellic acids are produced in the embryo and involving in the seed germination. How they are inducing the seed germination? So they are inducing the initiating the enzyme synthesis. Here some enzymes are synthesized. So these enzymes are converting the stored food materials into glucose, and these are involving in the respiration. So energy is produced. With the utilization of this energy, the seed is germinating. So like that here the gibberellic acids or gibberellins are involving in the seed germination. If the gibberellic acids are not produced during the seed germination, here no seed germination occurs. That's why these are the very important phytohormones which are inducing the seed germination. So these are the physiological effects of gibberellic acid or gibberellins. Next uses. Next uses of gibberellic acids. Here fruit growth. Mainly in the fruit growth. How the gibberellic acids are involving in the fruit growth. So mainly in the apple fruit, the size and shape. In apple, size and shape. So the gibberellins are improving the shape of the apple fruits, and these are also elongating the apple size. Next, grapes. Here grapes also grape stalks. If the gibberellic acids are present, if the gibberellic acids are produced in the plant body, the fruit growth or the stalk, fruit of the stalk. Generally in the on the common stalk, here number of fruits are arranged. So there are the here bunch of grapes. The grape stalks, fruit stalks. The fruit stalks are they are very short. Here the completely all the fruits are arranged compactly. If all are arranged compactly, here sometimes the grape bunch it is affected by fungal pathogens. Fungus affects on the here grape bunch. That's why here the gibberellic acids are improving the or elongating the grape fruit stalk or pedicel. So these are elongating in the size or in the lengthwise. That's why these are loosely arranged. If they are loosely arranged, no fungal pathogen effect. If they are compactly arranged, easily affected by the fungal pathogens. So here the grape stalk length is increasing due to the gibberellins. That's why these are loosely arranged. So the gibberellins are improving the fruit size, shape, and in the grapes stalks are also here, the size also or length is also increasing. Next, delay ripening. The gibberellic acids are here delaying the ripening process or senescence and abscission or old age or aging process and falling of leaves, flowers. These are also here delaying of ripening. It is also one effect of gibberellin. Next, malt yield. In the brewing industry or in the alcohol industry, so the alcohol is produced from different raw materials from barley or wheat seedlings or malt. Next, fruit juices. Mainly in the barley seeds are monocot seeds. Here the seedlings are used in the brewing industry or beverages. So the malt, so what is malt? Maltose is the sugar. During the seed germination, the stored food material is converted into maltose sugar. So the seed is germinating into seedling. The seed is germinating into the seedling. So after the seed germination, here the stored food materials are converting into maltose sugar. So this type of seedlings are used in the brewing industry. If the gibberellic acids are not present, the seed cannot germinate. So if the seeds are not germinating, we cannot utilize these seeds in the brewing industry or in the alcohol industry. So it is germinating due to the effect of gibberellic acids. So the stored food materials are converting into malt or maltose sugar. These seedlings are used in the brewing industry or alcohol industry for fermentation process. So mainly the barley seeds or wheat seeds are used. So here it is malt yield. In the malt yield, malt production also, here the gibberellic acids are involving. Next, sugarcane yield. In the sugarcane plant, generally the stem is the useful part. Stem. If the stem is very lean, it is very short, so the sugar content is also very less. If it is very lengthy and it is very thick, so more sugar is present in the or more sugar is stored in the stem. So here generally gibberellins are improving the or they are elongating the stem or internodal length. Right here the gibberellic acids are due to the effect of gibberellins, or if the gibberellins are sprayed on the crop, or if the gibberellins are produced in the plant body, here it is so the sugarcane stem. So if it is very lean, short, dwarf, so very less sugar content is stored, so it is commercially not beneficial. So if the gibberellins are sprayed on the crop, the stems are elongating in the size or in the lengthwise and in the widthwise or thickness. The thickness of the stem also increasing. So here more sugar is here storing, the stem is storing more food materials or carbohydrates or sugars or sucrose. So the sucrose is stored in the stem, so that is commercially beneficial. Here the gibberellins, without gibberellins and after spraying the gibberellins, with the use of gibberellins, if we see the difference, so with the use of gibberellins, here 20 tons of sugar, more sugar is produced in 1 acre. Without gibberellins, some sugar is produced, but if we use the gibberellic acids in 1 acre of place, the sugar plants are the yielding 20 tons of sugar, more sugar is produced. So that is the effect of gibberellins. Next, quicker maturity. Here the quicker maturity means here the gibberellins are involving in the maturation process. Here some plants are in the young stage or in the juvenile stage. Here the juvenile plants like conifers, gibberellins are involving in the quicker maturity. Here in juvenile conifers, young conifers are here they take so much time for the maturation period. So here if the gibberellins are used with the help of gibberellins, here the juvenile conifers they reach the maturity period very early, and early flowering and early seed production. So if the seeds are produced very early, so it is commercially beneficial. In economically, seeds, in some plants the seeds are very important economically or commercially, or they are edible. So in such type of plants, here the gibberellins are used. So they come to the maturity period or flowering stage. So early flowering or early maturity period, so we can see, and seeds are also produced very early. It is commercially beneficial. So it is about gibberellins and their physiological effects.